Welcome to the 2023 LACNETS Special Educational Event. I'm Kavya Veligapudi, Executive Director of LACNETS.
I'm Lisa Yen, Director of Programs and Outreach for LACNETS.
And I'm Heather Davis, the Associate Director of Advocacy and Outreach at LACNETS.
A special thanks to our speakers, our LACNETS team, and Rich at TVP Live for this high-quality program. Today's presentation is on novel translational research for metastatic NET patients at the NCI. We would also like to thank our sponsors, Ipsen, Advanced Accelerator Applications, ITM, and Lantheus. LACNETS was previously known as the Los Angeles Carcinoid Neuroendocrine Tumor Society. Now LACNETS is Learn, Advocate, Connect Neuroendocrine Tumor Society. While you often see Lisa and Heather leading the meetings and programs, we're led by a team that includes me as the Executive Director and Board Member, and Board Members Donna Gavin and Mary Dunleavy, who is living and thriving with NET. Now over to you, Heather, for some important tips about today's event.
Thank you, Kavya. Follow LACNETS on social media to stay up to date on our upcoming webinars and NET news. We're on Facebook, Twitter, and Instagram with the handle @LACNETS. And be sure to add us to your Safe Sender email list. I'd like to remind our audience that LACNETS webinars are created for educational purposes only and do not substitute for medical advice. The views shared in this webinar are personal opinions of our experts. Please contact your medical team with questions or concerns about your individual care or treatment. And now I'll turn it over to Lisa.
Thank you, Heather. I'm excited to introduce our speakers for today. Dr. Michael Yaffe is the David H. Koch Professor of Science and Professor of Biology and Biological Engineering at MIT, where he has been a member of the faculty since 2000. He is also an attending surgeon and intensivist at Beth Israel Deaconess Medical Center, where he specializes in injury and surgical oncology. He has been the director of the MIT Center for Precision Cancer Medicine since its inception. Dr. Yaffe earned his MD-PhD degree from Case Western Reserve University. He then completed a residency in general surgery at the University Hospitals of Cleveland and New England Deaconess Hospital and a fellowship in surgical critical care, trauma, and burns at Harvard Medical School. He was a postdoctorate fellow with Lewis Cantley in the Department of Cell Biology at Harvard Medical School. Dr. Yaffe is the scientific editor-in-chief of the peer-reviewed journal Science Signaling, and he serves on editorial boards of two other peer-reviewed journals, Molecular and Cellular Proteomics and Cell Cycle. In 2021, Professor Yaffe was elected to the Association of American Physicians, an honorary medical society dedicated to the advancement of scientific and practical medicine. He was also named a McVicker Faculty Fellow at MIT, an honor recognizing his teaching contributions at MIT. He is a decorated veteran of the war in Afghanistan and retired as a colonel from the U.S. Army Reserve Medical Corps in 2021. Thank you for your service to our country, Dr. Yaffe. Dr. Oliver Jonas is the director of the Laboratory for Biomicro Devices and associate professor in the Department of Radiology at Harvard Medical School with appointments at Dana-Farber Cancer Institute and the Broad Institute. His laboratory focuses on engineering approaches to precision medicine in cancer and other diseases. He develops novel technologies that enable the interrogation of disease in a more rapid, local, and precise manner. In addition to seeking clinical impact, he also aims to apply the technologies to generate fundamental new insights into biology, with a particular focus on drug resistance, an area in which he's published several original articles. Dr. Haidira Del Rivero is well known to the LACNETS community. Dr. Del Rivero grew up in Veracruz, Mexico, and after getting her medical degree from the University of Veracruz Medical School, she did internships in Mexico and then completed her internal medicine residency at New York University Laguna Medical Center in New York City before going to the NIH for her endocrine fellowship, followed by her medical oncology fellowship. Having training in both oncology and endocrinology is extraordinary and makes her uniquely suited to treat neuroendocrine tumor patients. Dr. Del Rivero is a leader in the NET medical community and serves on the board of directors for NANETS, the North American NET Society, which is the NET professional medical organization. She serves as the NANETS Guidelines Committee co-chair. Our LACNETS community knows Dr. Del Rivero to be a passionate and compassionate physician and advocate. She is a frequent speaker at both medical conferences and patient education events, and she also serves on our LACNETS Medical Advisory Committee. In October 2022, Dr. Del Rivero was honored with a Clinical Excellence Award by the Center for Clinical Research at the NCI at the NIH. Dr. Jonathan Hernandez is a surgical oncologist and hepatobiliary surgeon, and he serves as the chief of the surgical oncology program at the NCI at the NIH. Dr. Hernandez specializes in the treatment of metastatic and primary tumors of the liver, bile ducts, and pancreas, as well as in the surgical management of complex abdominal tumors. Dr. Hernandez graduated from medical school with honors from the University of Florida and completed general surgery training at the University of South Florida. During his residency, Dr. Hernandez spent 2 years at the Moffitt Cancer Center and Research Institute doing research on liver metastasis and trying to understand what leads to metastatic spread. Following residency, Dr. Hernandez completed fellowship training in both surgical oncology and hepatopancreatobiliary surgery at Memorial Sloan Kettering Cancer Center. During his fellowships, Dr. Hernandez spent an additional 2 years in dedicated basic research studying metastatic colonization. Dr. Hernandez has authored over 100 peer-reviewed publications and has contributed book chapters in numerous authoritative surgical textbooks. Heather and I had the honor and pleasure of touring the NCI and the Hernandez lab in October 2022. We have had patients in our support group seek care with Dr. Del Rivero and undergo surgery with Dr. Hernandez. It was amazing during our tour to see the behind-the-scenes look at what happens to liver tumors after they are removed. We were very impressed by their innovative work and all that they're doing. Dr. Hernandez said, "It isn't enough for me to remove the liver tumors. We have to find a way to cure this disease." I was literally moved to tears by the groundbreaking work and by their passion and dedication. We also wanted the patient community to get the same behind-the-scenes look that we had, so you too could be informed, encouraged, and inspired. We now welcome our esteemed guest who will share novel translational research for metastatic NET patients at the NCI.
Hello, everyone. My name is Jaidira Del Rivero. I am a medical oncologist and also a board-certified endocrinologist at the National Cancer Institute, National Institute of Health. First, I would like to give thanks to the LACNETS team for inviting us today to discuss our research in neuroendocrine tumors. I'm going to give an introduction of what we are doing and also how patients can be referred to the NIH. And then Dr. Hernandez and his team will discuss the exciting research that they're doing in neuroendocrine tumors. First, I would like to discuss the structure of the National Institute of Health Intramural Research Program. The NIH has about 27 institutes, and one of them is the National Cancer Institute. Within the National Cancer Institute, there are other funding entities, but in terms of the intramural program, we have the Center for Cancer Research, and that's where Dr. Hernandez and myself, we're part of the Center for Cancer Research. In the Center for Cancer Research, there are 21 basic research laboratories and 21 clinical branches. Within those clinical branches, there is the surgical oncology branch where Dr. Hernandez is part of, and also Dr. Michael Yaffe, who is the branch chief of the surgical oncology branch. And together they collaborate with Dr. Oliver Jonas, and you're going to hear from them later about what they're doing in terms of the research in neuroendocrine tumors. Myself, I'm part of the developmental therapeutic branch, and I also work closely with Dr. Nitin Roper, as well as Dr. Suresh Kumar, and the three of us were also working on different research models to advance the knowledge as well to develop better therapies for neuroendocrine tumors. Within the Center for Cancer Research, there is a diverse group of basic, translational, and clinical investigators. One of the goals is to understand cancer biology through different mechanisms: cellular molecular biology, genomics, tumor microenvironment, and immunology. Moreover, we also wanted to understand the rapid translation of basic findings into innovative clinical trials. Also, the Center for Cancer Research has a clinical trials programs for tumors with unmet needs. We also know that neuroendocrine tumors is an unmet need to develop better therapies and also to improve the quality of life for patients living with neuroendocrine tumors. As we all know, neuroendocrine tumors can be anywhere in the body. There is a unique group of tumors or neoplasia that can affect different organs, more commonly the small intestine, the pancreas, the lungs, but also other part of the body, such as the adrenal gland and other more rare areas. That said, even though we understand about neuroendocrine tumors better than what it was years ago, we still need to develop better therapies for these tumors. We can see here in the treatment landscape of neuroendocrine tumors that back in the 1990s, there was not that many treatment options. But now in the last decade or so, we can see that there is different treatments available for neuroendocrine tumors. That said, we still need to understand what is the sequence of treatments. And also, we still need to develop better therapies that can improve the quality of life of patients living with neuroendocrine tumors. Now, what are clinical trials? Clinical trials are research studies that test how well new medical approaches work in people. Through clinical trials, doctors find new ways to improve treatments and the quality of life for people with certain diseases. Researchers design cancer clinical trials to test new ways to treat cancer, find and diagnose cancer, prevent cancer, and also manage symptoms of cancer and side effects from its treatment. Clinical trials are the final step in a long process that begins with research in a lab. Before any new treatment is used with people in clinical trials, researchers work to understand its effect on cancer cells in the lab and in animals. They also try to figure out the side effects it may cause. So now we explained briefly what clinical trials is all about, how important is in the management of neuroendocrine tumors. However, we also facing with some challenges. We have challenges in the management of neuroendocrine tumors. Even though we have now a few FDA-approved agents, sometimes we need to understand when and what combination therapeutic approach should be used, what a specific type of neuroendocrine tumors may benefit from a specific treatment, and also the sequence of treatment options. Moreover, patients with neuroendocrine tumors and their families face unique challenges. And one of the reasons is because it takes a long time to diagnose, and sometimes the cancer is diagnosed in advanced stages. Most medical centers have limited experience with these tumors, and sometimes patients have to search for experts and sometimes travel outside their home region in order to see an expert, in this case, for neuroendocrine tumors. There's no standardized treatment guidance, and that creates uncertainty in a search for answers, and sometimes our patients feel isolated. Also, the NET researchers also faces some hurdles. Sometimes it takes a long time to accrue patients, considering this is a rare cancer. And also, there are multidisciplinary care needed for patients living with neuroendocrine tumors. And one thing is that, in one of the focus as well here, and what we do in the clinical center is that we're trying to develop preclinical models to then test different drugs and then take it to the clinic. However, we know that there's limited diversity of NET preclinical models, and thus is a challenging situation. Without these robust preclinical models, sometimes the drug development can be a little bit halted. So that's the reason why one of our goals here at the National Cancer Institute is to understand more about neuroendocrine tumor biology, develop preclinical models, and then with that, be able to develop novel therapies. Now, I am fortunate that I'm currently the principal investigator of a protocol. This is a tissue procurement and natural history study of neuroendocrine neoplasia. That means that includes, this protocol includes neuroendocrine tumors as well as neuroendocrine carcinomas. The objective is to characterize the natural history of all neuroendocrine neoplasm. And that data will include patient demographic, clinical characteristics, patterns of disease progression, response or lack of different therapeutic interventions, disease recurrence, as well as understand patients with neuroendocrine tumors, how long they live with certain treatments, to understand and guide us as well what would be a more personalized therapy for patients living with neuroendocrine tumors. Why natural history study is important? Researchers follow people with cancer over a long periods of time. So with this natural history for neuroendocrine tumors, will allow me to follow patients for long periods of time. We also collect details about patients' medical histories, tissue, sometimes blood, sometimes saliva, sometimes both, and as well as tumor samples. And that's when those tumor samples usually are obtained from surgeries or from biopsies. Researchers who conduct natural history studies use this information to study questions such as how do specific cancers form, growth, and spread? What genes causes cancer to develop at a high rate in certain families? We know that neuroendocrine tumors more commonly is sporadic, meaning we don't know the cause of neuroendocrine tumors. However, approximately 10% of these neuroendocrine tumors may be in families. We can also learn clues to help prevent cancer, and we can also learn clues to help develop new therapies. And that's one of our goals with the research that we're doing here at the NCI, or the National Cancer Institute. So how, in terms of clinical trials at the NCI and how patients can be involved or referred to our studies. Patients can contact us directly, and at the end of the presentation, you're going to have my email to contact me directly. There is also a Center for Cancer Research Referral Coordinator, and that's the phone number, but usually it's better if the patients or physicians contact us directly. How to take part of, we need to have a diagnosis confirmed by your physician. In this case, we need to have a diagnosis of neuroendocrine tumors. Have an illness that has been studied in a clinical trials that is accepting patients. In this case, Dr. Hernandez will discuss more about his research, but for me, the way I see patients through the clinical center is through the natural history study, as I described earlier, and there is also a consult protocol or screening protocol as well to allow us to see patients at the clinical center. Also, the research team for the trial that you have an interest to be involved will review all the records, and depending on the results of the review, then we may invite you to the clinical center for a screening visit, and if you have an interest to participate, we will discuss later what the options are. Once a patient is enrolled in a clinical trial, care at the clinical center is free. If you would like to obtain more information about the opportunities to participate on a clinical trial, there is a website on the bottom, www.cancer.gov, and please visit the webpage. Now, in terms of clinical trials at the NCI, specific for neuroendocrine tumors, either the patients or the referring physicians is referred to us. We collect the clinical information, we collect pathology, we collect imaging reports, we review all that information. We have a team here that specializes in neuroendocrine tumors, from surgical oncologists, and medical oncologists, endocrinologists, nuclear medicine, radiation oncologists, and so forth. Once we review all that information, then we provide an opinion to the patient as well as the physician. The patient can then be enrolled into the data collection of clinical care and interventions in the Clinical Cancer Research Center of the NCI. And that also allows, this protocol allows also for patients to have surgical interventions as well. If a patient that is coming to the clinical center and surgery is indicated, patients will be able to participate in the study, the clinical care. Moreover, we also offer other studies that the patients can be co-enrolled as well. And in this case, the natural history study for neuroendocrine tumors also allows us to enroll patients and follow patients over time. This protocol also allows me to see patients that are not eligible for surgeries, and Dr. Hernandez will discuss more in detail about his research and patients that are eligible for surgery. And as we know, patients with neuroendocrine tumors, even in the metastatic setting, surgery is still considered an option. And if you wanted to learn more information, LACNETS also have a series of videos and webinars and recorders that you can obtain more information about when surgery may be indicated in this situation. And Dr. Hernandez will discuss more in detail about his exciting research. Now, if the patient is not eligible for surgery, then the patients can be enrolled in my study. And also, if the patient requires treatment right now, we have a study, a treatment trial of Lutathera. We know that Lutathera is approved for the management of neuroendocrine tumors in combination with a pill called olaparib. And the idea is to make Lutathera more effective, and that's for patients with gastric and pancreas neuroendocrine tumors. We're seeing those patients, and we're currently enrolling those patients into the study. And we're also opening more studies, treatment studies for neuroendocrine tumors, as well as for neuroendocrine carcinomas within the next few months. Also, there's multiple clinical and treatment trials available at the NCI, and working together with your referring physician, we can find other treatment options available for your tumor type. Once the patient is eligible for surgery, patients can enroll in different protocols, as we discussed earlier, and including the natural history study for neuroendocrine tumors. Dr. Hernandez is our surgeon expert in neuroendocrine tumors. He specializes in complex general surgical oncology, as well as hepatopancreatic biliary surgery. And once the patient is eligible for surgery, there is going to be a piece of tissue taken for different research goals. And Dr. Hernandez, as I said earlier, the reason of this webinar is to discuss the exciting research of Dr. Hernandez and his team, but also my team in the developmental therapeutic branch. We're also working on different research aspects of neuroendocrine tumors to be able to develop models and develop novel therapies for neuroendocrine tumors, as well as to understand the genomic signatures and correlate with treatment response. And then this research may be a discussion for another time, but more importantly is to see what Dr. Hernandez has to share today after me. Now, if you have any questions, please contact us for help. You can see that there is different ways to contact by via phone or live chat, or also via email. You can contact me directly and through my email. This is my email. And as I said earlier, if you have an interest to be seen at the NIH, we collect all the information. You can send me an email directly to be able to collect all the information, and then we'll review and we talk with Dr. Hernandez as a team. I have to say I'm very fortunate here to be at the NCI, to be able to do the research that I love in neuroendocrine tumors, and to be able to work with fantastic researchers that wants to improve the quality of life of neuroendocrine tumors, such as Dr. Hernandez. With that, I would like to thank the LACNETS team, Ms. Lisa Yant, Ms. Heather Davis, Ms. Mary Dunleavy, Donna Gabend, and Kavya Velagapudi, as well to continue the legacy of Ms. Giovanna Imbesi, who has dedicated her life to improve the life of patients with neuroendocrine tumors. And with that, I wanted to thank you for your attention, and I am available for any questions with Dr. Hernandez. Thank you.
Hi, I'm Jonathan Hernandez. I am a tenure track investigator at the National Institutes of Health. I'm chief of the surgical oncology section and head of the metastasis biology section. I'm here to talk about our research efforts as they relate to neuroendocrine tumors. Before I do that, I think it's probably important that I sort of highlight how I see the disease process. And this is a video I had made highlighting a pancreatic neuroendocrine tumor. And what you can see are there micrometastatic cells or disseminated tumor cells that spread from that primary tumor. And this probably happens very early on in the process. And so most of the time when we see people who have developed evidence of spread, they probably have micrometastatic disease throughout their liver. Now, sometimes some of those cells will begin to grow and macroscopic metastases or gross disease will begin to develop. Now, this is a dotatate scan showing positive tumors in the liver for a patient with a neuroendocrine tumor. And so what has happened essentially is some of those cells that have left the primary tumor and ended up in the liver have begun to outgrow and form macrometastases. That also, but it's important to realize that though there are tumors that we can see, there are likely additional small cells that we cannot see. These are called disseminated tumor cells or micrometastases, such that somebody with a skill set that allows them to go in and remove the tumors that you can see, you are likely, very likely to be leaving disease behind. And so this is a complicated process. It's termed a metastatic cascade. And so, but it's important to realize though, just because we see only certain things on a CT scan or a dotatate scan does not mean that is all the disease present. And so that has to be realized by all of the providers involved in the care of these patients. All right, so those of course can be removed, but often what happens is the disease will come back over a person's lifetime, and then we're dealing with a potential problem that would be more difficult to deal with. But this is because those micrometastases that are present probably early on in the disease process, some of those have attained the ability to outgrow, and they've now formed, again, gross macrometastatic disease or spread, that we can see on an X-ray, CT scan, or a dotatate scan, for example. And then so this becomes, for a lot of patients, this becomes a basically, I like to, as an analogy, this would be a battle of attrition. And so we're here dealing with this, and we have tools, but it's important to define our goal, I think, and that's going to be continuing to battle as long as possible and as well as possible. This is going to go on for a lot of patients for many, many years. And so we have tools in our arsenal. Surgery and ablation are local, potentially regional therapies. We have peptide receptor, PRRT, with Lutathera and other agents. We have embolization. Potentially, sometimes we can employ radioembolization with Y90. We have some systemic drugs, lanreotide, everolimus, capecitabine, and temozolomide. These agents in our arsenal rarely get us to what we would like to get to, which is no disease ever comes back, which of course means we need to expand the arsenal. And so my task thus far has really been to paint the problem as I see it, but really the task is, how am I going to address this? How do I get us expanding? How do I expand our arsenal? And so this is where the research efforts come in. And as I see this, there are two ways to do this. I think, first we need to develop biologically relevant tools. Cell lines are not great. There are not a lot of them for this disease. I'll tell you, in other diseases, there are a lot of cell lines, and most of the time they have very limited translational applicability. The FDA just put out a statement that using mice to translate those findings to patients has been problematic, and we need better tools. And I would absolutely agree with that completely. But tools are simply tools, and so we need to then interrogate the tools. We need to use the tools to begin to understand what they're telling us and what they're telling us as related to drugs. And so I've done this in the following way that I'd like to show you. And so I view tumors as the following. They're not simply balls of tumor cells. They're very complicated. They have a lot of parts. There's a lot of dynamic parts to this. There are clearly tumor cells, but there are a whole host of other cells present in a tumor, including various populations of immune cells. There are fibroblasts around. There are some of the cells that would otherwise be native to the organ where the metastases is forming. There's this all-important matrix, so that the structure of the tumor is different than the surrounding liver. And all of these things make it really hard to employ any dissociative techniques such that you would try and pluck out the tumor cells only and begin to use them to try and sort of tell you what would happen in an intact tumor. So I think, that the dissociative techniques are not that they're not worth anything, but the translational applicability of all models that rely on dissociation are somewhat limited, and you'd like to have an intact tumor. This is just an image we captured of a tumor, and you can really realize the complexity of this. There's not one cell type here. And this is a high complexity thing. And so understanding how a very complex thing responds to a drug is probably very different than understanding how a cell line or something like that would respond to a drug. And so what I've sort of done when I first got here, it's been about 6 or 7 years now, is I said, I think it's going to be possible to remove a tumor from the liver and keep that liver alive outside of that person for a period of time. It's a complicated process. It's taken us about 5 years to develop a machine that's capable of doing this, but it is absolutely possible. And what I'd like to do is I'll show you that in a more detailed format. I'll have Jacob Lambden, a surgery resident in the lab, go through the machine with you part by part so you can really understand this and appreciate the complexity. But in essence, it is still a machine, and it is still keeping a piece of liver, and it can be a segment. So we've been able to do this with small parts of livers. We don't need to cut out half the liver. But even then, you still have a sizable piece of tissue containing usually more than one tumor in it. And so the question becomes, what questions are you going to ask of this? And so we've developed these pillars, what we call pillars of interrogation. Some of these are live dynamic imaging, and Tracy Poo, another surgery resident in the lab, will go through our efforts related to live imaging, and that can be done on biopsies on the liver straight from the patient or from our liver ex vivo machine as we've been giving drugs to the liver outside of the person. We can dissociate the tissue and do it flow cytometry to understand cell populations better. Of course, we can apply lots of omics. That would include transcriptomics, but most importantly, proteomics and metabolomics to begin to understand what the metabolic and signaling state of the cells are. And of course, because we have this perfusion of the patient's, some of the patient's own blood, some of it is blood from the donor bank. We can look at biomarkers in the perfusate to look and see what correlates with response. We think this is going to be sort of very valuable to begin to understand what drugs are actually doing in real time. That is a very difficult. The fourth dimension, the time dimension, is extremely difficult in all preclinical models, but we have the ability to do that. And of course, it would depend on the agent we're looking at, but we would look at agent-specific evaluations. And so what I'd like to do now is have Jacob Lambden and Tracy Poo show you our ex vivo liver machine and show you some of our pillars of interrogation as they relate to live and dynamic imaging. So we would begin to develop a full picture about the tumors.
Hey, my name is Jacob Lambden. I'm one of the surgical oncology research fellows here at the NCI. I work in Dr. Hernandez ex vivo lab for the past 2 years, and this has been my project, constructing this machine, which we call the TBOM, which stands for Tumor Bearing Organ Machine. Before we get into all the parts and how we intend to use this to help patients, I'll give a little background into what motivated us to develop this machine. So really, if you were going to study cancer, the ideal way to study cancer would be to study a human tumor inside of a human patient. But if you're talking about doing things like planning surgeries or testing experimental drugs or knowing which drugs work best for an individual patient, all of those things, if you did it in a human patient, would subject that patient to potential harm or toxicity from those experimental kind of things. In order to circumvent those problems, researchers, will study cancer cells in a dish, or we can study cancer cells in an animal, like a mouse. But ultimately, the trouble is that human beings aren't cancer cells in a dish, and human beings aren't mice. So when you're looking at which therapies are going to work for a human being, it doesn't always translate from those other models to the clinics. So the idea was, what if we could take a human tumor inside a human liver and take it to the patient to the operating room and resect that tumor like we normally would as part of their standard of care. But then instead of sending that tumor off to the pathology lab, what if we could take it and trick it into thinking that it's still inside the patient? Then if it thinks it's still inside the patient, it would behave as if it were still inside the patient, and we could do all of these experiments on that tumor without subjecting patients to any toxicities or any harm or any risk. So that was the motivation. So the guiding principle behind our machine development was going to be rebuilding the body around the tumor in order to trick the tumor into thinking that it's still inside the body. So that's what we did. And now we can walk through sort of our mechanical organ systems, starting here with our heart, which is these two pumps right here. So we have two pumps, one for the portal vein and one for the hepatic artery. These are the two inflows to the liver, which you have in a normal person. These pumps need to spin at different speeds to give different pressures, different flows. The artery needs to be pulsatile, the vein continuous flow. All of this stuff is very complex and particular, and it's needed to help replicate the physiology that goes on inside a person if we want the behavior of the tumor to reproduce what it does inside a person. So our next artificial organ system we have is our lungs, which is these two things right here, which are oxygenators. So we flow gas across these one way and blood across them the other way. And in doing so, it allows us to deliver oxygen to the blood, which we circulate through the organ, and remove waste products like carbon dioxide. After the oxygenators, we have a kidney. And this is our kidney right here, which is an improvised dialysis circuit, which we constructed, which allows us to remove waste products from the blood, which are generated over the course of keeping this organ alive. After the kidney, our next organ system would be our pancreas. And in a normal person, the pancreas is responsible for keeping the blood sugar in normal range. So what we have here is a glucose sensor, which a patient would normally have implanted in their arm, which senses the blood sugar of the blood. That information is relayed back to our computer, which then tells our syringe pumps, which inject insulin and glucagon, which are hormones that keep the tight regulation of your blood sugar levels. And that's automated based off of the levels detected in the blood. All right, so now we'll step over here and take a look at the star of the show here. So this is a piece of liver with the patient's tumor still inside it. You can see here that the liver rests on this perforated hammock, which allows us to capture any blood which spills during the process. And then into the vessels of the liver, so the portal vein, the hepatic artery, and the hepatic vein, we've sewn cannulas, which allows us to bring blood into the liver and out of the liver the same way it would be in the body. And then we also have a cannula sewn into the bile duct, which allows us to drain the bile which the liver normally produces.
Hi, I'm Tracy Poo, and I'm a surgical oncology research fellow at the National Cancer Institute, NIH. I work under Dr. Jonathan Hernandez in his lab exploring ex vivo models for metastatic tumors. What does that mean? So we take a tumor from the patient in a clinically indicated operation. We take part of that tumor to pathology, and we take part of that tumor back to our lab to be able to better characterize this in our lab space, to be able to not only see the tumor after it's gone through pathology and normal ways of interrogation, but actually seeing the relationship while it's alive inside the body. So our target is to actually keep this tissue alive and be able to image it. So my project is using 4D live imaging. So basically trying to take a video of this tumor and its relationship between normal liver and a tumorous origin. So for this project for neuroendocrine tumors, as you know, patients can live with this for multiple years, but often patients present with multiple different liver lesions, and these tumors actually can take up most of the volume of the liver. But we don't really understand why these slow-growing tumors are so aggressive, and we don't understand its relationship with the normal tumor liver tissue within the body. So that's how 4D live imaging can help us better understand this interaction at a microscopic level. So here, by using our lab's 4D live imaging using this confocal microscope, we can actually take a lab sample and put it into our microscope. And then from this, we can see on a cellular level the relationship between this tumor and the normal liver tissue. So using 4D live imaging, we have our laser colors that denotes what we're seeing on the screen. So as you can see on the bottom of the screen, we see a lot of green. So green denotes our tumor cells or tumor markers. We can see that blue is interspersed everywhere because blue denotes DNA within the nucleus of cells in both tumor cells and normal cells. But we see that there's no green on top and it's mostly just blue, so we can deduce that this is normal liver tissue up here. And then the other interesting relationship we can see from this video is that the cyan small balls that are rolling around within the tumor tissue, that's actually our immune cell marker. So we can see a lot of these immune cells, or these tumor infiltrating lymphocytes, attack the tumor cells within the microenvironment. And we can see that some of these immune cells are moving slower while some of them are moving faster. We can actually see some of these immune cells really engaging with the tumor markers and actually attacking the tumor itself. So now that we understand where the immune cells are in our 4D live imaging in 4D space and how fast they're moving, we can actually use this response to target drugs and drug treatment and actually make drugs that we know that are existing that work, understand not only how they work, but how to make them even better in the next iteration.
Okay, so I think at this point you probably fairly well understand how we're able to keep livers with tumor in them alive outside of people. And I think you probably have begun to understand that we've been developing these pillars of interrogation for some time, and that some of them are fairly advanced. But we have to get to a goal. Again, our goal, as we sort of laid it out in the beginning, was we need to expand our arsenal. And I think it needs to expand on two fronts. The first is, can we delay or prevent the outgrowth of micrometastatic disease? That has been and remains, at least in my opinion, one of the most difficult things to study in oncology, because it's not like we can see or capture these cells. So we've devised a plan to do this. And this plan looks like the following. So when we have a piece of liver, and this again can be a segment or something small on the machine, and it has a macrometastases in this, we can put labeled antibodies into the system and begin to detect these micrometastases. They will be revealed by their cell surface expression of things we can identify. And then these biopsies can be interrogated, because we'll have, if you think about this in terms of a needle in a haystack, we've essentially cut down the haystack. We've made it much smaller such that we're able to then identify these cells. And so as a first approach, what we're doing is dissociating these biopsies or pieces of ostensibly normal liver. And we're using flow cytometry to best refine our markers to capture the tumor cells and only the tumor cells. Again, this would be an ostensibly normal liver. So this is a patient who has, metastatic disease in multiple sites, and we would believe that that person has also micrometastases, of course. So when we take out some of the tumor in normal liver, we would be looking in the normal liver and identify what these tumor cells look like. And then ultimately, we've devised a plan to perfuse that liver with a dual labeled set of antibodies. This will be dual labeled with fluorescence and radio labeled, such that they can be retrieved. The issue with the liver is that there's a lot of autofluorescence. So fluorescence can sometimes be challenging, but the radio signal will allow us to hone in on certain areas. And then we would then slice this liver up. And we won't be using a single surface marker. We want orthogonal data points so that we can identify a cell based on multiple markers. We can say with confidence that is in fact a tumor cell. Again, this would be we would have a radiation signal, which can be placed on basically a reporter plate, and we can identify areas of radiation, which can then be cored out. And then we will basically make our haystack very workable at this point. And we can begin to understand, are those micrometastases simply spread throughout the liver, or are they in very certain areas, for example, along the collagen surfaces of the inflow vessels and the capsule and the outflow vessels? What are their immune microenvironments like at these very early time points? Is there anything we can use to sort of keep them asleep and not actually allow them to outgrow? I think these are extremely important questions. And I think we've devised a plan to answer this, and we put this plan in action. We're still on the flow cytometry parts of this, but I think it's going to be really important. Next, I think in expanding the arsenal, what we would want to do is we want to ultimately bring efficacious agents to patients. And that's, of course, patients who have macrometastatic disease. And then that begs the question, how are we going to do that?
Hello, I'm Dr. Michael Yaffe. I am the director of the surgical oncology program at the National Cancer Institute, and I am the David H. Koch Professor of Science at the Massachusetts Institute of Technology, where I direct the Center for Precision Cancer Medicine. And what I'd like to tell you about very briefly today is the future of precision cancer medicine for neuroendocrine tumors. Now, as I suspect many of you are aware, the traditional dogma has been that cancer is primarily a genomic disease, a disease in which specific mutations in specific genes lie at the root cause of cancer. And therefore, understanding the genes that are mutated in cancer should, we have been told, indicate the best treatment. And as part of that promise, we have been told repeatedly that genomics, that is, the sequencing of the DNA within tumors, is the answer to cancer. In this paper from Nature last year, 38 different tumor types were characterized with the expectation that the identification of mutant genes in different tumor types would be directly relevant to our understanding of how to treat those particular tumors. And this promise has delivered in some, but not all tumor types. Here I'm showing you the case of colorectal cancer, and I'd like you to focus your attention on what I'm showing you within this red box. Every row of this red box indicates a gene, and every column indicates a tumor from a specific patient. And I hope you can notice that all I need is 3 or 4 genes to be able to characterize the different mutations that are responsible for causing colorectal cancer. And understanding these gene mutations tells me, at least in part, in theory, all I would need to do to be able to treat this type of tumor in these different types of patients. The problem here, of course, as I suspect many of you are aware, is that this promise of cancer genomics has completely failed in the case of neuroendocrine tumors. The slide is busy, but I will try to walk you through it. Every row that you see here is a different gene. Every column is a neuroendocrine tumor from a different patient. And I hope you will notice that compared to colorectal cancer, where every patient had mutations in one of only 3 or 4 genes, here in pancreatic cancer, we have a desert. The vast majority of patients have very few mutations. Some patients have absolutely no mutations, and there does not seem to be any single gene that is responsible, by and large, for the vast majority of neuroendocrine tumors. This is obviously a huge disappointment. But in fact, this shouldn't surprise us, as indicated in this commentary that was published in Wired magazine in 2009, Beyond the Genome. And I'll just point out that in Wired magazine, their comment was that when scientists finished sequencing the genome, the answers to diseases like cancer were just supposed to follow. And this, which was published 14 years ago, already said that the promise has gone unfulfilled and that genetics just isn't that useful for understanding tumor biology. Why? Why is it that genetics is not useful? And the reason is because what genetics gives you turns out to be a very intriguing parts list. Genomics tells us the parts, but it doesn't tell us how those parts are connected together. This is an example, if I can use electronics as an analogy to the cell, of what genomics gives us. Genomics tells us the parts list, but I would challenge any of you in the audience to tell me whether these parts belong to a VCR or a television or a cell phone. In fact, all we know from this parts list is what there is. We have no idea how these work together. This is what genomics gives us. Unfortunately, what we need to cure cancer is something more like this. We need to know how those parts are connected together and how that interconnection is failing. In fact, if we go a little further with this analogy between electronic and biologic circuits, what we really want to understand is not what the parts are, but how the parts are connected and how in normal cells and in cancer cells, the signaling is flowing through the wires in this circuit. And in biological circuits, the wires turn out to be protein kinases, and these carry the current of signaling. Protein kinases are what control how different components of the cells talk to each other. What we really need then, in order to understand cancer, is a way to measure the signaling current that's flowing through a cancer cell and figure out why that's different than in a normal cell. Now, in electrical circuits, we have a way to do this. We can use something called a probe card, where we put our circuit on the probe card and we read out the voltages and currents all distributed throughout the circuit. And then we know what piece is broken and we know what piece we have to repair. The problem is we can't do this from cancer genomics in cells and tumors. And that's why I wrote a decade ago in this editorial that the future of precision cancer medicine is to fill in the missing data, not just the genetics, not just the genomics or the RNA expression, but the signaling state and the proteomics of cancer. Because if we combine what we know about the genomics with a much deeper understanding of what's wrong with the signaling in tumors, particularly neuroendocrine tumors, then we'll have a better idea of what the tumor-specific vulnerabilities are and what kinds of chemotherapy or other treatments we should use to treat this particular tumor type. Now, you might wonder, if dysregulated signaling is so important in cancer and cancer treatment, why has everyone focused so intensely on cancer genomics? In fact, I wrote just a few years ago an editorial, and I said, why did geneticists steal cancer research if cancer is primarily a signaling disease? And the reason turns out to be because it's very easy to get genetic information, and it's very difficult until now to get signaling information, even though the signaling information is what we need to treat these types of tumors. Most of the signaling information that we get uses a technology called mass spectrometry. And what mass spectrometry does is it tells us about the state of proteins within normal cells or cancer cells or any cell type that we want to interrogate. And out of this, we get thousands and thousands of pieces of data. But until now, we have had no understanding of how to take that data about the proteins and understand what it was telling us about how that circuit was wired together. I'm happy to and that's because what we really need is a Rosetta Stone. The Rosetta Stone, you probably recall from your social studies class, was a stone that translated ancient languages into more recent types of language like Greek. And that allowed us to decipher hieroglyphics because we could speak the language through the Rosetta Stone. What we really need to be able to understand the signaling state of tumors is some type of Rosetta Stone for cancer. And if we had a Rosetta Stone, we could understand what my friend David Janes calls the dark phosphoprodium. We could understand the proteomics of neuroendocrine tumors in a way that would allow us to identify what was wrong with these tumors that seem to lack clear genetic drivers of cancer. The problem has been that we've been vastly ignorant of the vast majority of protein kinases and their substrates. And most of the work that's been done on protein kinases has focused on a subset of kinases called tyrosine kinases. Most of the focus, as I said, has been here, even though most of the action, particularly in neuroendocrine tumors, is here. It's on a different group of kinases that control that signaling state in tumors, and those are protein serine and threonine kinases. But until now, we have not been able to understand how to understand the signaling state of the cell using these kinases and proteomics. Interestingly, everything has changed over the last 5 or 6 years. And this just was revealed in a paper that we published today in the journal Nature. What we have now been able to do is to build the Rosetta Stone that we need to be able in the future to decode the signaling state of neuroendocrine tumors and hopefully steer us better onto a path for treatment. Why do I say that? So this is from today's MIT News. And what's described here is an atlas of protein kinases that's going to help us decipher the cell signaling pathways that hopefully should yield new cancer drugs that we could use to treat a wide variety of tumors, including neuroendocrine tumors. Here's that kinome tree that I showed you before, and I mentioned to you that we had no idea for the vast majority of protein kinases shown in the colored parts of this tree what their substrates were. And what I'm showing you now is work that took place over the last 10 years between Lou Cantley's lab and Ben Turk's lab and my lab that has now decoded the signals that come from each of these kinases. What this means is that we can now use mass spectrometry to profile, based on those peptides that we get back, what protein kinases and what signaling pathways are blocked and what signaling pathways are overactive. And by understanding the signaling state of the tumor, now we can begin to devise therapies that might be useful.
Thank you very much for having me today. My name is Oliver Jonas. I'm an associate professor at Harvard Medical School and excited to present to you our lab-in-a-patient microdevices for multiple drug screening in cancer patients. The unmet need that we're seeking to address relates to therapy selection and drug development in oncology. As many of you will know, there are many drugs in clinical oncology to choose from. There's about 150 or so different approved agents on the market. They're often given in combinations of 2 or more, and these are very expensive drugs. They often have toxic side effects, and it's usually not clear which drugs are going to work in any given patient. And so what often happens is that patients are tried on a standard of care first-line treatment. We have to wait weeks or months until we know if that treatment was effective. And if not, we try a second-line treatment or maybe a third line later. And of course, that leads to a delay for the patient. It leads to more tumor resistance over the course of time, and it leads to toxicity and a significant burden for the healthcare system. So the promise of precision medicine, and one that we are seeking to bring about, is to be able to test all of the available treatments right at the start, and to then choose the one treatment or combination that's going to be most effective for that specific patient. A related unmet need exists in drug development. So the vast majority of drugs that are ever tested in patients fail, and most of those fail later in development, when a lot of resources and time have already been expended in developing them, and they fail due to a lack of efficacy. So being able to know much earlier in the drug development process if a given treatment would be effective, and to define the patient population in which that treatment may be the most effective, would have significant value. The technology that we developed to address this unmet need consists of implantable microdevices that are placed directly in the tumor and can measure the response of the tumor to up to 20 different treatments at the same time. These microdevices have 20 different reservoirs. Each reservoir can have a different drug or combination of drugs, and is placed directly in the tumor using a standard biopsy needle and remains in the tumor for about 3 days. During that time, each of the drugs are released into separate regions of the tumor where they're able to react with the native tissue. We then extract the device with surrounding tissue. This could either happen at a second biopsy or at the time of surgery. And then we process the specimen by fixing and paraffin perfusing it, and then sectioning through that specimen to get single cell layers that are cross sections of this device tissue specimen, where we have tumor, and then we have the device in the middle, and we have separate regions of the drug, like the ones that are shown here in blue and pink, where we can then measure how the tumor has responded to each of those drugs. And we use a range of techniques that I'll talk about more later. The most important things here are, one, that we can test many drugs at once, because we test them spatially separate in non-overlapping regions so that we can see what each drug does individually. Each of the drugs are delivered at nanodoses, so they're very, very small doses that are non-toxic to the patient, and allows us to find predictive biomarkers of response, so that we can test both new drugs and approved drugs and compare their efficacy so that we can see what the effect is within the patient's tumor. One important aspect of drug activity always is the dose. So for many cytotoxic agents, which are currently the majority of the standard of care, the more drug you give, the more effective tumor killing you will get. But of course, that comes with toxicity. So it's very important to know how much drug we actually need to administer to a patient in order to get the maximum anti-tumor effect. And one of the nice things about our approach is that we have a gradient of drug release, meaning that we have different concentrations that depend on how close we look to the device reservoir. And then as we get further away from the device, we have lower concentrations. And so we're able to measure the tumor response as a function of concentration, meaning we can understand how much drug do we have to administer in order to kill, for example, all the tumor cells within a given region. And that's very important clinically, and can help patients not only get the most effective treatment, but also be able to get that at the lowest possible toxicity. One other important aspect of the measurements that we make is not just how the tumor cells respond, but also how the other cells within the tumor, the immune cells, the fibroblasts, the stromal cells, how they react. There's a lot of interest in immunotherapies. There's a lot of promise. We've had some really great success stories there, a lot of really good responses where patients have essentially been healed by immunotherapy, but it's still a small percentage of patients that are actually benefiting. And so one very promising field of research is try to combine immunotherapies with other drugs like targeted agents and chemotherapies that essentially will kill the tumor in such a way that immune cells are attracted to the site where the drug is released, and then we can combine them with the right immunotherapy to get an even greater effect. And that's what some of these techniques are promising to do. So we're able to measure not only the direct cell kill effect of each treatment on the device, but also how these drugs are reshaping the tumor microenvironment and which combination treatment in terms of immunotherapies we could then use to get an even greater effect.
At this point, you've heard from two collaborators of mine. One is Dr. Michael Yaffe. The other is Dr. Jonas from Harvard. And you've now understood that what Dr. Yaffe has been able to do is he's basically built a Rosetta Stone for us, such that if we take phosphoproteomic data, he can tell us which of the kinases may be relevant, and interpreting that data informs us about signaling. Now, signaling leads us back to kinases because that's how the cells communicate within the cell. There's signaling circuits, and tumor cells steal and circumvent normal governances to have aberrant signaling. And so understanding what cells may be driving that signaling may reveal what we want to be able to target. And Dr. Jonas has a device that we can put into tumors, which can be loaded with kinase inhibitors and drugs that would activate the immune system. And obviously, these combinations are particularly interesting to multiple cancers. I'd like to bring that to neuroendocrine tumors. And this is the way I envision doing this. So a patient comes, and this is the trial we're bringing to fruition, and a patient comes with multiple liver metastases, and these are, I can either cut all these out or I can debulk nearly all of their disease. But before I do that, about 3 days before, we would put Dr. Jonas's device into these tumors, and these would be loaded with kinase inhibitors in conjunction with immunotherapy agents alone. Cape-10 can be loaded into this device. Everolimus can be loaded into this device. such that, this can be then correlated with what actually may happen to that individual patient. Anyways, after the 3 days later, I would go in and remove all of these tumors. And what we would then do is we would look at the tumors that had the devices in them and the other tumors as well. And we would look at a phosphoproteomic profiling to understand kinases of interest. We would do immune tumor profiling of the microenvironment to understand the immune components that are at play. And then we would determine, then in the context of Dr. Jonas's device, we would determine tumor responses to the various kinase inhibitors in the context of the data we got from the phosphoproteome. And we would do the same sort of thing. We would determine the tumor sensitivities to the combination of kinase inhibitors and immuno-oncology agents in the context of the immune profiling and the phosphoproteomic profiling. I think this is going to be a huge step forward for us. And I think what we're going to see is we're going to see patterns of commonality among certain patients depending on their profiles. And I think, for somebody who operates on fairly aggressive metastatic disease, I feel it an obligation that we offer something more. And I think this is going to be that something. Of course, if somebody requires a segment of their liver removed, we would put it on the machine, but this is going to give us drugs that would likely work. And so we would then try combinations ex vivo. And so I think this is the avenue forward for patients with metastatic disease from neuroendocrine tumors.
Welcome to our Q&A session. I just want to thank you both for your time to take on some questions today. And I also want to give a shout out to your fellows, Dr. Hernandez, Tracy and Jacob, and their dedication and hard work. I don't know if it came across in the presentations how dedicated they are, and what they shared with me and what you shared with me in terms of how they babysit the tumors and sleep on a cot, taking care of them. So if you want to speak a little bit more to that and their work.
I am definitely blessed with a very dedicated team. Surgery residents we're sort of used to that sort of thing where we stay up long periods of time, but it's done not out of obligation. It's done out of commitment and belief in what we're doing is going to matter. But I definitely benefit from having surrounding myself with great people, of course, Dr. Del Rivero included.
Yes, thank you. So let's get into the questions. There are several questions that came in. And one very pertinent question is, how and where can neuroendocrine tumors be donated for research? So Dr. Del Rivero, let's start with you.
Yes, thank you so much, Lisa. And first, I would like to thank you for the opportunity to be here today and discuss what we do, and especially with Dr. Hernandez. As I said earlier, I feel very blessed to have him and as the surgeon for neuroendocrine tumors. And as you can see, his research is really exciting. So I was just sending him a text earlier to, oh, I love every single one of things. So that said, I can start, and then maybe Dr. Hernandez can also comment. If the question is whether you can donate any neuroendocrine tumors to the NIH. So there's different protocols. I think I mentioned earlier that the NIH has what we call the clinical cancer research has 21 basic labs and clinical labs, and within those different branches as well, there are different protocols too. I can tell you in our branch, we do have a protocol that we can accept neuroendocrine tumors tissue, as well as somebody wants to donate future tissues or body as well. That said, we like to mention that there are different institutions as well in different hospital like UCSF and Memorial Sloan Kettering and all different hospitals. They have different protocols how they can procure tissues too. It's always important that you communicate as well with your treating physician. What are your goals? And especially when you're trying to plan for the future as well, I think it's important to have that discussion not only with the family but as well with the treating physician. So how you want to provide that. I have to say, I always tell my patients, this is when you want to do this, I feel like this is very novel because you're also helping the community and to answer so many questions in neuroendocrine tumors as well. You advance the education and science as well. I think it's important always to ask those questions to your trained physician, and there are different institutions that you're able to donate tissues for neuroendocrine tumors as well, and not only here at the clinical center, and maybe Dr. Hernandez can explain the process in his branch. We have different protocols where we can accept that within our range, but at the same time, many different institutions are able to accept that as well.
Dr. Hernandez, I mean, your work in the ex vivo liver machine is very impressive. And what if a patient's like, hey, I want my liver on that machine? What would you say?
I would say that we can probably make that happen depending on a number of things. But I think about it in terms of sort of fresh tissue and the things we can do with fresh tissue, and then there's things we can do with paraffin embedded tissue. And so, if a patient wants to be seen by us and wants to have treatment at the NIH, that person would be a candidate for us to do some of the cutting edge stuff that you saw earlier in the video, including live imaging, including ex vivo perfusion, assuming a number of parameters are met. And I think the stuff we do with live tissue would the patient would have to be treated at the NIH. There are other things that were not mentioned in the video that we would be able to do from paraffin embedded tissue. And so I think the questions are a little separate, but there are a number of good institutions around the country who really care about the disease. And I think it's important to learn from every single patient. I think that's really important. And I've always been sort of taken aback by how much people really care about not only themselves, but about the community in general, and about sharing tissue and about learning so others can benefit from their experience. And so again, there's a couple of institutions around the country that do very good stuff, and everybody's been collaborative insofar as sharing what we learn. But as far as donating tissues and things like that, I think the live tissue sort of stuff we're doing has to be, we have to care for that patient and do the operation here. The other stuff, there are things we're doing, particularly looking at the single cell microenvironments of liver metastases, which can be done off paraffin tissues, which can be shared between institutions.
Thank you for that clarification and also for that collaborative spirit. We really like to hear that from the patient community. And another question that is somewhat related, Dr. De Rivera, can you comment on whether or not it's possible to donate your body to neuroendocrine cancer research after passing?
Yes, I can comment on that, and maybe Dr. Hernandez as well. And I think that maybe a little bit on the question, kind of like follow-up question that was asked earlier too, that in how you're able to donate tumors. And like Dr. Hernandez said, there's different ways, whether if a surgery is done here at the NIH, if we can obtain tumor from paraffin blocks, meaning that usually when the tumor is resected, it sends to pathology, they store in pathology, and sometimes it can, depending on which institution it is, it can keep the tumor for a certain number of years, usually about 10 years. But again, every institution is different as well, and that's kind of like how we're able to collect different tissues too. So in terms of these questions, is also based on different institutions and what protocol they have, and we do have a protocol here. It's called the rapid autopsy protocol. It's part of our range, which allows us to bring patients after passing, and we want to answer different questions, and there's a protocol to do that. We usually take care of any transportation going here into the NIH, and that's something that usually we have a discussion with the family as well as with the patients and understand what her wishes are or the wishes of the patients are. And that's why I was saying earlier, it's very important that you always have a discussion with the family, a discussion with your physician, always having your wishes, and always respect the wishes of the patients as well. I don't know, Dr. Hernandez, do you have anything like that in your range? Or
We have the rapid autopsy program. I don't know how much we utilize this, because I think there's a lot that goes into end of life issues. Probably a topic for a different day. But if somebody's very interested in that, we certainly have that opportunity available. I know Sloan Kettering has a similar type program. These options exist, but there's a lot that goes into this, and families, it's possible, but I think a lot of things that conversation should be had with the care providers, early on, and I think as long as everything's planned appropriately, and that's the patient's wishes, then I think we can make that happen.
Thank you. Another follow-up question: how long are tumors stored at hospitals? If they take, for example, approximately 7 sections and use only 4, can they use the remaining for research for after X number of months? And what if a patient passes away?
Do you want me to start, Dr. Hernandez? Maybe you can,
Yes, I
at
think
least
you
for.
probably know more about this than I do, but hey.
Yes. So yes, I mean, I think I was mentioning a little bit about this earlier. So every hospital has a different program, how long they can store their tumor. Usually it's about 10 years. That's what my understanding is. I mean, in our protocol, we are very good. The research nurses are very good for asking what we call unstained slice and or paraffin block. So, and that's usually one of the patient usually allow us or give permission in order for us to collect that. For the purpose of our study, or at least the natural history study, what I can tell you is that we usually ask for about 15 to 20 unstained slides. And the reason why we ask for unstained slides is because we can do our own analysis, but not just that, we can always extract DNA, we can extract RNAs for future analysis as well. Sometimes it's better to obtain a paraffin block. So basically, it's in a small piece of tumor embedded in a paraffin block. It's easier because then we can do the cuts here at the NIH. We can obtain more tissue for further analysis. Sometimes I have to say what we have encountered is that when it's more than 10 years, the DNA is the tumor is not the same. Maybe we're able to make a diagnosis or maybe correlate a diagnosis, but we will not be able to obtain or extract any DNA or RNA to do future analysis. But yes, every hospital is different. My experience is like about 10 years. They started in other hospitals. I know that these neuroendocrine tumors, some of our patients have surgery more than 10 years ago. And when that happened, sometimes what we do is if there is a tumor that is available or accessible, we may obtain a biopsy. As we know, a lot of these neuroendocrine tumors can go to the liver. The liver, and maybe Dr. Hernandez can comment for, is an area that can be easily biopsy, that we can do biopsy and is easily to locate. And that's something that we do, especially if we wanted to correlate the diagnosis, if we want to correlate what is the Chi-67, if we wanted to do future analysis, we can certainly do that. Now here at the NIH, we studied tumor for a long time, more than 10 years. That's my experience, but I don't know what, Dr. Hernandez, what are your thoughts about it?
It's not uniform. So for example, let's say I take a tumor out and it comes out at 10 o'clock in the morning, that can go into paraffin and be processed right away, and that specimen's probably pretty good. If it comes out late in the day, a lot of times the pathologist protocols are such that it can sit overnight before it gets embedded in a paraffin block, and that specimen, as you can imagine, could degrade and other things can happen to it. I think it's highly variable. Um, but for the most part, these blocks stay good for a long period of time. And certainly we like to keep tissues for a long time because, as time moves on, you find out new things and you can then go back and look. And, at that time you have corroborative clinical history to give credence to your hypothesis. For example, if you were looking for a certain, correlative protein or whatever, I think the archival samples are really important.
Right.
There's some heterogeneity insofar as their quality. I would say the technologies are evolving that allow us to do more and more with paraffin embedded tissues, and I would say there's definitely things that are available now that I never thought would even be possible. And so I think
Yep.
these samples are valuable for a number of reasons because they have the value of time. Like, we know what will happen. There are some patients who are going to do very well and some patients who are going to do less well. And that gives us sort of stuff you wouldn't otherwise know that without the time variable. So I do think they're important, and
Right.
Yeah.
Can I add a comment on that? And I completely agree with Dr. Hernandez, and I think that's something very important that is added on our protocols. Whenever there is a patient that is enrolled in a protocol, there is a consent, and on that consent, at least on the natural history study, we ask if we collect tissue from surgery, are we able to store the tissue for future analysis? Because I think we're learning, right? We're learning, we're asking new questions. Medicine is evolving, the management of neuroendocrine tumors is evolving, and if we wanted to ask future questions, I think, like Dr. Hernandez says, it's very important to go back, store the tissue, go back and ask new questions. At the same time, the other question that I ask on the protocol is, can you allow me to share your tissue with other investigators, not only within the clinical center, but also outside of the NIH? Because these are, I think I always say, collaboration is key. To understand more about these neuroendocrine tumors, and we are fortunate we collaborate also with other institutions, the same with Dr. Hernandez, and sometimes putting all these samples together, putting all our brains together, so we can definitely ask questions that can definitely change how the management of neuroendocrine tumors. And I'm really hopeful and excited about this and how we are. As you want to ask me, how you feel about neuroendocrine tumors, I feel very hopeful right now because I think we're understanding more. We have researchers like Dr. Hernandez who really wants to understand more about these tumors and finding a cure for these tumors, and excellent surgeons. So I'm very excited for what's about to come.
Oh, thank you for that, Hope.
oh,
There's
thank you.
There's this question, Dr. Hernandez, specifically for you and your drug delivery device. If you could elaborate on the drug delivery device implanted into tumors, assuming there's a response to the drug or combination of drugs, would you assume that intratumoral delivery would be equal, superior, or inferior to venous interarterial delivery? And then if you get a signal, what are the implications for treating the patient?
Yeah, this is an excellent question. And so I think all of these sort of ways to gauge drug responsiveness have limitations. And I like this device developed by Ali Jonas. It has advantages. And the advantages are there can be 20 drugs in each of those devices. There can be combinations. They don't have to be FDA approved because they're really micro dosing. One of the limitations, I would say, which is pointed out by the question, is, well, you're sticking this device straight into a tumor. You're going to get drug where it may not go. Is that physiologically relevant? Excellent question. It certainly raises questions about drug delivery and these other things that I think are, again, problematic, but everything is going to come with pluses and minuses, and everything is going to have sort of limitations. One of the reasons I like the ex vivo liver so much as a model is the intact, all of the irregularities, the abnormalities associated with the circulation, the circulatory system, the microvasculature, it stays intact and the same as it was in the person. So we can circumvent some of that. On some level, you should think about this device as a high throughput screen that needs to be validated in a different way. And this gets to the, part of the second part of your question is, what will you do with the data? We've talked about this, and the easy answer would be we would anything that looked really good, we would give it to the patient. The truth is, doing that is not straightforward. This requires FDA approval, particularly if they're not FDA approved drugs. But we've discussed writing single patient protocols if we were to see somebody who had a really good response to, let's say, a combination of agents, combination A and B, and that person had gone through all clinical available options to them. There were really no trials left that they sort of fit for, and they were progressing. We could definitely write a single patient protocol and propose using those drugs so long as we had some dosing idea about what to give. And so I think there is a way to translate that back to the person, their single patient protocols. It would be hard to write a blanket protocol, at least in the beginning, because we need to see how this is going to work. But I do think there is an opportunity, and I do acknowledge that this one of the limitations of the device is, are you really going to get that kind of drug delivery when you shove this thing straight into a tumor? I certainly had reservations about that in the very beginning, but I do think it's good data. All of this stuff is valuable. We will learn something from it, and don't forget now that there have been new delivery systems set up. Paclitaxel would be one. Nab-paclitaxel Abraxane is approved. That's a combination of paclitaxel to get it better to where it needs to go. And so there are ways we can do this sort of stuff, but we would need to know the agents. And so I think all limitations acknowledged, I think this device is a pretty good idea.
Yeah, thank you. There are many questions, and unfortunately we can't get to all of them as our time is running out, but let me just take this question. How does someone get seen at the NIH, and what's the follow-up after someone is seen at the NIH? Dr. Del Rivero, let's start with you.
Okay. Okay, good. As I said earlier, I mean, first, we need to have a protocol or that of the tumor that we would like to study. As I mentioned a little bit earlier in the presentation, we do have a natural history study for neuroendocrine tumors. That protocol allows me to see patients and follow patients long term. We have a consult protocol as well, that if the patient, they have, sometimes it happens that some of my neuroendocrine tumor patients, they are very happy, with their treating physician, and they just want a second opinion. So we have a consult protocol to do that. We also have a standard of care protocol, and in that protocol is Dr. Hernandez are able to see surgeries as well. But within the protocol, if a patient comes to the NIH and have surgery, they can co-enroll on different protocols. Even my natural history study, Dr. Hernandez, what we call biospecimen collection too, where he can answer other research questions as well, and other protocols within the clinical center. I think the most important is that we have a team here at the NIH that we want to see those patients, that we want to understand more about these tumors and be able to develop effective therapies. And also depending on I say protocol because that's how the patients are seen here at the NIH. So like, for example, my natural history study allows me to follow patients for at least 10 years. And then with that, we answer specific questions as well and correlate as well with different outcomes too. That said, I always tell my patient, once you're part of the NIH family, we're here for you, and we're able to answer any questions, even if it's a consult protocol, standard of care, natural history, screening protocol. So we are here for you. Once you visit at the NIH, I tell my patients, you're part of the NIH family. We're always here for you. We're always able to answer any questions. Please don't hesitate to reach out.
Do you have anything to add with that? How does someone get seen at the NIH, and what's the follow-up?
Well, this is where I greatly benefit from working with Dr. Del Rivera and all her expertise. Our sort of system is that the patients come through her and her team, and we meet once and now seemingly twice a week to go to review patients, go through scans, and make decisions about things. And then I would say that's step one. And once we make decisions, one of us will reach out and speak to the patient directly, go through their history. Go through things, and I like to, if it's someone for a potential operation or just wants opinion about their scans, I'll go through the scans with somebody, render opinions about things, and it sort of evolves from there depending on the patient's interests, wishes, and so on and so forth.
Thank you. And let's close with this last question. What words of hope would you offer to the NET patient community? Dr. Del Rivero, let's start with you, and then we'll close with Dr. Hernandez.
Yes, I like this question, and the reason why is because, as I said earlier, I mean, I feel hopeful. 10 years ago, we didn't have the treatment options that we have today. We still have many unanswered questions. And I feel by understanding what we do in the work we're doing and collaborating also with other NET experts in the field, we should be able not to only improve the quality of life of our patients, but at the same time, hopefully one day develop a cure. That's what we're always hopeful, right? And like I said earlier, I'm very fortunate to work with Dr. Hernandez. No, he's an excellent surgeon, an excellent person, and is just, I always feel very blessed whenever we discuss cases with him. He gives a different perspective about these neuroendocrine tumors. He asks questions, but more than that, he doesn't give up in his patients, and neither do I. So, and I think we work very well together. Both of us, we really want to develop better therapies for neuroendocrine tumors. And I think that we're here for you, and we're hopeful. And because of you, you are our passion. Because of you and all the patients, we're here for you, and we love what we do. And so that's kind of like one of the things I say to my patients. I'm grateful to my patients every day, and I'm grateful for everything that you do. You are our passion, our inspiration, and you are the reason what we do and what we do. And so do never give up. We're with you, and we're going to continue fighting with you.
Wow, thank you for your care and commitment. Dr. Hernandez, what about you?
Yeah, I'm not sure I can follow that very well, but I'll do my best. I would say this is an incredibly exciting time. I think we're on the very beginning of expansion of therapies and better options for people. And I think that's going to come in it's going to come through research, and it's going to come through our better understanding of the tumors. And, I think what Dr. Yaffe said about sequencing, the world has been a sequencing dominated thing for cancers for some time. And I think only recently have we begun to understand how to decipher the signaling and the efforts that, the signaling that occurs within these tumors and the phosphoprodium, and how do you sort of map phosphopeptides back. And this is all possible now. And so where we were a sort of a one-track kind of thing for a while, I think another avenue is really about to open for us. And I think, some of the work that Heidi's doing with organoids and some of our work are going to allow us to really take advantage of this information. And so I'm incredibly hopeful. I think things, of all the diseases I treat, I think we're on the precipice of really moving in the right direction, and that's super exciting. Um, I'm extremely encouraged, and I would echo Heidi's comments about doing everything we can, and we don't give up, and we will continue to do whatever we can to help people. I feel that's a privilege and an obligation, and that is this is what we exist for at the National Institutes of Health. We exist on taxpayer dollars. All the research is funded by you, by the patients, people who pay taxes, and all the work goes back to you, and it's really important. And so everybody at this institution is like 100% in. This is what we do. This is why we come to work. And when you start to see things move like this, it gets people even more excited. And I think we're at a very hopeful time.
Wow, thank you so much, both of you. I mean, your curiosity and commitment, dedication, it's very encouraging, I could say, as a caregiver myself. And just seeing all the work being done behind the scenes, it's really encouraging to know people are fighting for us, opening those doors and creating possibilities. And those possibilities will hopefully lead to, better treatments and outcomes and better quality of life for all of us. Thank you so much for all that you do. And we just, from the bottom of our hearts, thank you.
Yeah.
Thank you.
Thank you, Lisa. Thank you, LACNETS.
Thank you, Dr. Del Riviero and Dr. Hernandez. We want you to know that you are not alone. To this end, LACNETS offers many programs and resources, including our weekly virtual NET support group, our monthly caregiver-only support group, NET Vitals, our NET communication tool, and a health and wellness coaching program. We encourage you to go to our website, lacnets.org, to find out more information and resources. LACNETS also has a monthly podcast featuring experts who answer the top 10 questions in their field. Our latest episode is on the key considerations for NET surgery with Dr. Alexandra Ganji from Cedars-Sinai. We encourage you to take a listen. And now I'll hand it off to Heather.
Thank you, Lisa. Follow us on social media to stay up to date on our upcoming events and NET news. Our handle is @LACNETS. We want to thank our valued sponsors, Ipsen, Advanced Accelerator Applications, ITM Pharmaceuticals, and Lantheus. Again, our sincere thanks to our distinguished speakers, Dr. Hernandez and his dedicated team, Dr. Del Riviero and the National Cancer Institute. our technical director, Rich at TVP Live, and everyone for joining today. We'll see you here again on the 7th.